So brief review. You see DNA is a helix. And then DNA helicase comes along and separates the strands. And I don't think this metaphor is going to work. Maybe I'll just put the ferrets in. Ferrets. No reason, but here are ferrets. Okay, metaphor failure aside, let's move on to the next subject. Transcription and translation. In transcription and translation, we see what we do with DNA. Because we know that DNA are the instructions for you. But now we need to know how we go about using the instructions. What good are instructions if you can't use them? So, when you use instructions, you are expressing genes. So gene expression is using DNA. So in gene expression, you go from the instructions from DNA or the gene to a protein. Because that's all a gene encodes is protein. So there's some important terms we're going to be using during this chapter. Transcription is one. Transcription is making RNA from DNA. When you think about transcribing, right? When someone transcribes a book, think of an old monk before the printing press. Books had to be copied by hand. They would transcribe a book, and they would use the same language when they were transcribing. They just make a copy. Well, transcription is making RNA from DNA. And we're still using the same language, nucleic acids. It's just one of them is slightly different. It has a uracil. It's RNA. So we're making an RNA copy of a DNA gene. So making a copy of a gene out of RNA. And then there's translation. This one's easy. When you translate a book, you take it from one language and convert it into another language. Well, when you translate RNA, you take it from the language of nucleic acids and translate it into the language of proteins. So you are going from RNA to protein. And both have some pretty cool mechanisms for how it works. So why do you do it? Well, with proteins, they can be enzymes, they can be hormones, they can be involved in cell signaling, they can be involved in the structure, they can be involved in your immune system. Your antibodies are proteins. Pretty much everything is involved with proteins. The synthesizing of certain proteins at certain times is why you are the way you are. You're just the product of a cascade of proteins being produced in specific amounts at specific times. That's it. And so transcription and translation is gene expression. It's often called the central dogma of biology, that you go from DNA to RNA to protein. Because almost all life, in fact, all cellular life, follows the central dogma. If it's a cell of any kind, prokaryote, eukaryote, archaean, protista, human cell, whatever, they all go from a gene made of DNA to an RNA copy of that gene to a protein constructed using the instructions in the RNA copy. So let's look at the role of RNA in all of this. The nucleus is a very crowded area. You have like 10 feet or 6 feet of DNA wrapped up, tightly packaged, shoved into the nucleus. Very, very crowded space, all right? You do not want all of your protein synthesis to just be crammed in there. So, one of the roles of RNA is to take the instructions from the nucleus to the cytoplasm. In fact, that RNA copy of a gene is messenger RNA, or creatively abbreviated mRNA. RNA can also be a component of a ribosome. The ribosome is made primarily of RNA. The ribosome is the site of protein synthesis. In other words, the ribosome is where mRNA goes to be translated into protein. So, what do you suppose we call RNA destined to be ribosomal? Ribosomal RNA. How would we abbreviate that? That's right. The pirate's favorite RNA. RNA. Yeah, leave me alone. That was funny. And then finally, there's RNA responsible for taking amino acids from the cytoplasm to the mRNA at the ribosome. So we can transfer amino acids from the cytoplasm to the mRNA strand at the ribosome. And this this RNA that transfers is this transfer RNA would be abbreviated as, that's right, tRNA, or phonetically terna. Okay, maybe you don't want to go with phonetic. Life is written in your DNA. Like I said, the instructions for everything is in your DNA. The instructions for your proteins are in your DNA. Proteins are how you express genes. So genes are everything. Making proteins starts with DNA. So now let's talk about transcription, making proteins. So, well, I already said transcription is making a copy. So a DNA transcription is making an RNA copy out of DNA, and that is your messenger RNA. And the players involved in transcription, the enzymes, are transcription factors, super creatively named, and an enzyme called RNA Polymerase II. RNA Polymerase II is a fantastic molecule. Whereas in DNA replication, you needed DNA helicase to unwind things, and then DNA polymerase to add nucleotides on there, as far as you guys are concerned. But as far as my 111 students are concerned, you needed DNA helicase, single-stranded binding proteins, RNA primase, DNA polymerase I, DNA polymerase III, ligase. You needed a lot of different enzymes to do everything required to make a single copy of DNA. So you guys are lucky. All you need to remember for replication is helicase and the general DNA polymerases. When we look at RNA polymerase II, it's like a home shopping network combined item right there. You know, when they sell their juicer, pocket knife, you know, car door opener, seatbelt cutting, I don't know, the home shopping network devices, right? RNA polymerase II does everything that giant list of enzymes I rattled off does. It binds to DNA, it unwinds the DNA, it exposes the template strand, it builds an RNA strand, and it puts the DNA back together. That is a big list of things to do for a single enzyme. It's pretty epic, honestly speaking. So how does it do its job? Well, there's a couple of steps to it. Step one is to find a promoter. A promoter is a region of the DNA that basically tells the enzyme, hey, the gene starts right after me. Scientists call it the ta-ta box. So, step one is for RNA polymerase to find the ta-ta box. Stop giggling, woman. You're old and mature.
Not really.
Shh. The ta-ta box. The ta-ta box is ahead of the start site. In other words, just before the start site is the ta-ta box. So RNA polymerase II finds the ta-ta box. And then it's able to find the start site. Step 2 is for eukaryotes only, and it involves those transcription factors. And these are molecules that aid polymerase, RNA polymerase II, in binding to the ta-ta box and the start site. And then step 3 is when RNA polymerase II just does its job. So RNA polymerase II binds at the ta-ta box. Then it senses the start point and begins making RNA from the template strand. So whereas in replication we used both strands as a template, in transcription we're only using one of the two strands of DNA as a template. So synthesis begins and elongation occurs. So basically in elongation, synthesis goes like this. RNA polymerase II separates the strand and it reads a nucleotide on the template. And say it reads an A. Okay, well, it takes the RNA nucleotide that's the base pair to that and adds it to its growing chain. So if it reads an A first, it grabs a U because A is the base pair of U. Because remember, in RNA, instead of A to T, G to C, we have A to U, G to C. Because uracil replaces thymine in RNA. So, it reads the nucleotide, says, okay, that's adenine, I'll put a uracil on here. Cranks over to the next one, guanine, all right, I'll throw a cytosine on. And it just keeps cranking down the chain, just reading each nucleotide set, bam, 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 putting down the complementary nucleotide, and that is elongation. Starts off with a single nucleotide and then just starts making a chain based on the nucleotides it's reading. So, puts down the RNA companion. Elongation is when it makes the chain longer. Basically, elongation is read a nucleotide, add the RNA companion. Read a nucleotide, add the RNA companion. Blah, blah, blah. Over and over and over and over and over again until it's time to stop. So it builds the chain from the 3' end of the DNA, which is honestly something we didn't talk about at all, so ignore that bullet point. Hooray! So you can think of transcription occurring kind of like trucks in a convoy. DNA polymerase, when you get on there, what am I trying to say? I'm trying to say that a single gene can be very long. It would be very time-consuming if you were only allowed to do one RNA polymerase II per gene. Instead, because RNA polymerase II is putting the DNA back together behind it, as one so if this is my start point right here, and the gene goes this way, here's my first RNA polymerase II, and it comes in and it starts reading it. And then this one comes in and it starts reading it. And then this one comes in and it starts reading it. And so they're moving along, and then they're kind of like, here's truck 1 and truck 2 and truck 3. They're in a little convoy traveling along a highway. And then finally you get to the point at which you need to stop. Termination. In bacteria, it's very easy. Termination of transcription in bacteria, very simple. There is a DNA terminator sequence. A DNA terminator is a metal endoskeletal structure with live flesh on it, allowing a robotic cyborg brain to hunt down Sarah Connor to prevent her from having John Connor and fighting Skynet. That's the terminator sequence. Okay, or it's a couple of sequences in the DNA that tell polymerase to detach. But I kind of like the terminator sequence, right? There's a DNA terminator sequence. I'm the terminator. I will terminate the polymerase. I'm just going to shoot the polymerase off the DNA with the gun and it goes. I'll be back. Right? That's the Terminator sequence. And once the polymerase detaches, the mRNA is released and it's ready for translation, the next step. So bacteria, very easy. They have the DNA terminator sequence. So hopefully you can remember that with the awesome Arnold Schwarzenegger voice with the Terminator. Unfortunately, you won't be able to use the Terminator movie reference for eukaryotes because eukaryotes are a little different. Instead of a terminator, they have the polydenylation signal. Polydenylation signal, however, can be memorized with some Arnold Schwarzenegger because Arnold Schwarzenegger did a little movie called Conan the Barbarian. And in Conan the Barbarian, one of his most memorable lines was, "Allah!" Right? He was thrown into some pit as the movie started with him turning some kind of, flour mill for no particular reason for 20 years. And then he was like Buff Arnold. And they threw him in a pit. And some guy jumped in with daggers and attacked Conan and he slashed him. And Conan's like, I don't know what's happening, but that hurts. Allah! And that's the polydenylation signal. AAU, AAA. Allah! So there you go. Those are some mnemonics you can take to the bank. So basically, downstream from the signal, the transcript is cut. So instead of the polymerase detaching, the mRNA is cut off of the growing RNA sequence. So the polymerase stays there and it detaches from a separate process. But the transcript is released when AAU, AAA appears in the RNA sequence being produced. Once that AAU, AAA is there, the signal is cut downstream and it releases the transcript. And that gives us pre-mRNA. Because, in fact, we're not ready for translation yet. Because eukaryotes just gotta be difficult. So here is RNA polymerase, my fancy guitar pick here. And it is separating my DNA strands and closing them up behind it. So here it is, closed together in front of it and closed behind it. And when it's acting, we separate the strands, exposing my template strand, this other strand being ignored. And we are adding RNA nucleotides onto the template strand, and then they're being cranked out over here. So I got a nice video for you, so let's watch together.
Transcription is the process by which RNA is assembled from a DNA template. The two strands of the DNA double helix are held together by bonds between complementary base pairs. DNA encodes genetic information in the sequence of bases along one strand. The portion of the DNA molecule that is a single gene or coding region is bounded by termination and promoter sites. A molecule of RNA polymerase binds to the promoter site. It moves along the DNA, separating the two strands of the double helix. Each now unpaired base will bind to a nucleotide in the vicinity which has the appropriate complementary base. In the synthesis of RNA, uracil is the nucleotide complementary to adenine. This process ceases when RNA polymerase reaches the termination site. The DNA strands bind to one another once again as the new RNA molecule moves away. This RNA is a copy or transcript of the message contained in the gene.
Ah, hooray. Now we understand 10 times better. Or you better. Anyway, so eukaryote DNA at this point, or eukaryote RNA, the transcript that we created, if it's in a eukaryote, is not in fact ready for translation. We have yet another step to go, unfortunately. So we have to do a little bit of editing. One of the first things we do is add the 5' cap and the poly A tail. 5' is just a number that is telling scientists which side of the DNA we're looking at, the 5' side or the 3' side. So I'll give you one guess which side of the DNA strand the 5' cap goes on. That's right, 5' side. Amazing. And then there's the poly A tail. Who wants to take a crack at, did I just say DNA when I was talking about the 5' cap? Bad, John. Bad. Talking about RNA. If I said DNA, ignore me. Okay, so then we have the poly A tail. Poly A tail is added on the other end, the 3' end. And I'll give you one guess as to what nucleotide you find most in the poly A tail. Did someone say adenine? Correct, sir. Your deductive reasoning skills are fantastic. So what's the purpose of the 5' cap and the poly A tail on our RNA transcript? Well, they protect it and they identify it as self, right? Because a eukaryotic cell is very large and very complex, and it has defensive mechanisms in place to prevent bacterial or viral DNA or RNA from hijacking the eukaryotic cell's needs. So if there's RNA floating around inside a eukaryotic cell that does not have a 5' cap and a poly A tail, that RNA is chopped up into little bits and not used. So the 5' cap and the poly A tail protects our RNA transcript from itself. Then the only other thing left to do is edit, because eukaryotic RNA transcripts are made off of eukaryotic DNA genes, and eukaryotes have extremely long genes compared to prokaryotes. A prokaryote gene tends to be significantly shorter. A eukaryote gene, many times longer. The problem is that just because a eukaryote gene is longer doesn't mean that all the nucleotides in that gene directly relate to the protein being made. In fact, eukaryotic genes have coding and non-coding regions all in there. And so the coding region, of course, translates into the stuff used to make the protein. That's what we want our mRNA to have. We want our mRNA to be made of, complementary base pairs to the coding regions. So we're going to have coding regions in our RNA transcript, but because the RNA transcript was a straight copy of the DNA, we're also going to have non-coding regions in our RNA transcript. In order to make it useful, we're just going to have to find a way to ditch the non-coding regions. The non-coding regions are called introns. I-N-T-R-O-N-S. Introns. It sounds like a faction in a Transformers movie. Optimus Prime! The introns are back! Not the introns! Quick! Notify other Transformers that I don't know the names of. That sort of thing. Now, the coding regions, which are left behind, are called exons. So the whole idea of RNA processing is to remove introns, leaving behind only exons. So the coding regions, the exons, are spliced together after we cut out our introns. It's kind of a neat process. Here, this dark green is an exon. So here's an exon on this side, and here's an exon on this side. And then this light green is the intron, the non-coding stuff. And we want to get rid of it. So we have these little molecules here called SNRNPs. They're SNRPs. That's literally what scientists call them, SNRPs, which is kind of adorable. You don't have to memorize how this works. It's just neat because the SNRPs cause the intron to fold together like this, and then they cut this is cut right here. The exon and the intron are cut loose from each other on this side, and then that's folded together, and then it's cut here again, and then we splice together those exons, and we cut up the intron and recycle the nucleotides from it. And once you have added a poly A tail, a 5' cap, and you've cut out all the introns, leaving only exons spliced together, you have eukaryotic mRNA. Functional mRNA in a eukaryote. Because eukaryotes are jerks. They can't just be like prokaryotes. Because when a prokaryote finishes copying off a gene, it's done and it's mRNA. Eukaryotes got to do all this extra junk. Ugh. So, the mRNA is messenger RNA, and that message is protein instructions. Part 2, we're going to take that messenger RNA out of the nucleus and into the cytoplasm in the next video.